Welcome to the next lecture on the cells of the nervous system. We're going to start by talking about the cells that nobody really talks much about or thinks about when it comes to the nervous system. Yet they're the most numerous cells within the nervous system, and without them, we would not be able to have our nervous system survive. And these are the neuroglial or glial cells. The glial cells provide structural support. They provide kind of the framework that gives our central nervous system its shape. They also protect the environment for our neurons. Neurons are extremely fragile, and many things can act as toxins to neurons. Our glial cells are responsible for maintaining the right environmental surroundings for these neurons. They make sure that the fluid is the right consistency and contains the right kinds of chemicals and ions and things to keep everything where it needs to be. The pH, all of that kind of stuff is controlled not by the neurons themselves, but by the glial cells. As I mentioned, they are the most numerous of all the cells in our nervous system. It's been estimated that there are as many as 50 times more glial cells than there are neurons within the nervous system. The other thing about glial cells is that glial cells can undergo mitosis. They can divide and create new glial cells. In most cases, neurons are not able to do so. There are 5 kinds of glial cells within the nervous system. There are astrocytes, microglial cells, ependymal cells, oligodendroglial cells, also known as oligodendrocytes, and Schwann cells. Astrocytes are the most abundant glial cell we have. These are some of the most important glial cells. If you want to say one's more important than the other, actually they're all extremely important. But astrocytes have a lot of jobs. Most of our glial cells have 1 or 2 primary jobs. Astrocytes have several different jobs that they perform within the nervous system in order to keep it healthy. They cover capillaries with what are called end feet. So if you picture your body as being like the cell body of an astrocyte, and then you have your arms and your legs coming off of your body. And if each of your feet and your hands were to kind of wrap around a capillary or to cover a capillary, that's kind of what astrocytes do. They have a lot of appendages coming off, and they have these what are called end feet, which are basically kind of like flattened endings on these appendages that cover capillaries up. And what they do is they help to regulate the endothelium of the capillary in forming and in the maintenance of the blood-brain barrier. So you have endothelial cells within the capillaries that are very tightly joined together, and these special junctions are helped to be sustained by the astrocytes. So if astrocytes were to break down or to become ineffective, we would find our capillaries getting to be very leaky and allowing things through from the blood into the brain tissue itself. And many of those things would be neurotoxic. There's a link that I provided you here, which has a good image of the end feet of a particular astrocyte. I think there's one, might be 2, but I think there's just one in this image. But it shows how those end feet wrap around a capillary. Other things that astrocytes do besides help to regulate and maintain the blood-brain barrier include to regulate ions and transport water. Having the right ion concentration is critical for having nerves function normally, which we'll see in a future lecture in this unit when we start talking about information transmission. These glial cells are also the main site for energy, for glycogen reserves, for storing glycogen, and for breaking down, for building up glycogen and breaking it down for energy sources. They're also very important at synapses or junctions from one neuron to the other where they talk to each other, because there is excessive neurotransmitter that gets put out in causing the communication from one neuron to the other. And the astrocytes help to absorb and eliminate this excess of neurotransmitter so that we have the right amount of information being transmitted from neuron to neuron. They help to not only remove excessive chemical out of the synapses, but they help to form and maintain synapses themselves. And they produce neurotrophic factors that are important for neuron survival. Neurons need certain kind of special chemicals to help them survive, and astrocytes produce some of these chemicals. Here's an image of an astrocyte. You can see it's got lots of appendages, not just 2 arms and 2 legs like we have. But many of them. You can see all the nuclei in the bright blue. I just think that's a pretty picture, if nothing else. But you have the one main astrocyte here shown in the front, and then a bunch of appendages. This one does not show an astrocyte by a capillary, however. Next we have our microglial cells. They're called microglial because they're very small. So they're micro because they're tiny. These guys are the Pac-Man of the central nervous system. They're phagocytic. They are the garbage men. They go cleaning up, taking up debris and things that have come from damaged cells, or simply cells that have started to wear out, or waste products that have been eliminated out of neurons. Also, if there's any kind of an injury, they will migrate along with astrocytes, will migrate to the area of injury and surround the affected area and help prevent the spread of toxins from the injury to surrounding areas. So they help limit the extent of the injury. And then the microglial cells start to slowly digest and engulf any dead or neurons or their parts. Ependymal cells are important for the production of cerebral spinal fluid. So CSF is cerebral spinal fluid. These cells are found within the ventricles of the brain and the central canal and the spinal cord, and they work with capillaries and the endothelium of the capillaries to form a special complex called the choroid plexus. And this choroid plexus is what produces cerebral spinal fluid. So you have a plasma filtrate from the capillaries, but then the ependymal cells modify it. So it's not just, they don't just filter the plasma as it's passing through, but they actually change the chemical composition to create the cerebral spinal fluid, and that is then secreted into the ventricles of the brain, and then it will flow within and surround the brain. And we'll talk about that more again in a future lecture. These ependymal cells have cilia on their apical surface. The apical surface of a cell is the surface that is exposed to the opening of a vessel or to the air. So for instance, in the case of the ependymal cells, it would be the side that's facing into the ventricle. If you were looking at a cell on one of your skin cells, the apical surface would be towards the air, so to the outside. If you're looking at a blood vessel, it's the side that's facing the lumen of the blood vessel. So that's what apical is when we're talking the apical surface. And in this case, we want to be able to circulate our cerebral spinal fluid So that apical surface of these cells has cilia on it. Cilia act like little hairs that move. They typically wave in one direction to help move things along in one direction. And here you can see ependymal cells, and you can see the little hair-like cilia along the edges. The major area that's blank at the top of the image, that would be the lumen of the ventricle. Our oligodendrocytes are what produce myelin. Now, in actuality, myelin is not a product of the oligodendrocytes. Myelin actually is oligodendrocytes. So oligodendrocytes, again, have a cell body, and then they have lots of appendages coming off this cell body. Each appendage wraps around the axon of a neuron and wraps around it several times. It wraps around it so tightly that it actually squeezes out all of the cytoplasm. So all the cytoplasm gets pushed out of the appendages into the cell body itself. And where you have this tight wrapping of just membrane, that's all that's left, everything that was in there got pushed out, where you have this tight wrapping of the cell membrane, that is myelin. A single oligodendrocyte will wrap around one or more sections of multiple axons. So you'll have myelin on a single axon is actually made up of several different oligodendrocytes. You do need fat to form myelin, and so if someone has malnutrition, they will not have the appropriate myelin. And this is particularly important when first myelinating in infancy, because you have to myelinate so much. Once we get older, you are constantly replacing myelin, but you're not trying to myelinate the entire nervous system. But as an infant, if an infant does not have enough fat in their diet, that will cause neurological concerns because they cannot adequately form myelin to insulate their nerves. Now, oligodendrocytes are wonderful because of the myelin increasing nerve conduction velocity. It allows information to travel more quickly along the nerve. However, oligodendrocytes also put out some chemicals that actually impede axon regeneration. So if an axon is damaged, it makes it less likely that that axon is going to be able to regrow. And one of the things that does that are some of the chemicals put out by the oligodendrocytes. Here we see a picture of an oligodendrocyte, and you can see they're showing 5 appendages, 3 going to one neuron, 2 going to a different neighboring neuron. But each of these appendages wraps several times around a neuron, squishes out all the cytoplasm, pushing it back up towards where the cell body is located. Schwann cells do the same thing, only in the peripheral nervous system. Oligodendrocytes are found only in the central nervous system, and Schwann cells are found only in the peripheral nervous system. Couple of differences, though, between Schwann cells and oligodendrocytes, besides their location, is that Schwann cells only wrap around 1 or maybe 2 or 3 axons, whereas oligodendrocytes will wrap around several. The last picture only showed 2, but an individual oligodendrocyte will wrap around multiple axons. But a Schwann cell, 1, 2, maybe 3, but that's about it. The other big difference is that where oligodendrocytes put out different factors that can impair axon regeneration, Schwann cells actually facilitate axon regeneration. So they help to assist our axons to regrow when they're damaged. And as a result, we're much more likely, if you have an injury to a nerve in the peripheral nervous system, to have that nerve regenerate than you will if with the same injury if it's in the central nervous system. And here again we can see now some Schwann cells, and because they only wrap around a single axon, the cell body is actually just, the whole thing is kind of flat and squashed. And in the little picture in the circle at the bottom right hand corner, you can see the cell body with the nucleus there, the nucleus shown in yellow. And it's all just kind of squished flat and wrapped itself around this axon. So now let's talk about neurons. These are the guys that get all the glory. They're the ones that are really doing a lot of the talking and transmitting information from one area to another. The cell body of a neuron is called the soma, and the processes that extend from it are called dendrites and axons. At the ends of axons, we have axon terminals, also known as synaptic terminals. We have afferent neurons in our peripheral nervous system and efferent neurons. Afferent neurons carry information to the central nervous system from the periphery. Efferent neurons take information from the central nervous system and transmit it to the periphery. So our afferent neurons are going to bring sensation into the central nervous system. Our efferent neurons are our motor neurons that are taking motor information out to the muscles. We also have interneurons that are neither afferent or efferent. They're neither motor or sensory, but they help connect the two of them together, and they connect other neurons within the central nervous system as well. So basically, interneurons are neurons within the central nervous system that go between two other neurons. The soma of the neuron contains the nucleus, which of course has the DNA. Many neurotransmitters, or chemicals that are used for communication between neurons and between neurons and other cells, are produced in the soma. Some of them can be produced in other areas, such as down in the axon terminal, but most of them must be produced in the soma, especially if it is a protein or a peptide. If the soma is damaged, this will always result in the death of the neuron. Dendrites are the main location for incoming information into the neuron. Dendrites are multi-branched. They're like kind of like a tree. In fact, they're called dendritic trees, and they have lots of branches, and on these branches they have smaller branches that are called spines. And it is on these spines in particular that we have synapses taking place. Now, synapses can take place on the dendrites and their spines, on the cell body, and on the axon terminals, but the majority of synapses take place on the dendrites and their spines. The number of dendrites, as well as the number of the dendritic spines, increases when you learn something new. This is called dendritic arborization. Again, it's called a dendritic tree. If you have a group of trees, you have an arbor, and that's why it is called dendritic arborization when there's increased number of branching taking place. The good news is that you can continue to learn and to arborize even into your 9th decade. So individuals in their 90s are continuing to learn all the time. Lifelong learning is not just a figure of speech. Next, we have axons. Axons can be either myelinated or unmyelinated. In truth, unmyelinated is not quite the case because there is a very thin layer of myelin, but it's like having a oligodendrocyte or a Schwann cell just wrap once around the axon, and there might be bigger gaps between them. But we call it unmyelinated. So some axons are myelinated and have a whitish appearance because of the myelin. Others are unmyelinated, and this creates more of a gray appearance. So when we talk about gray matter and white matter, it's letting you know if we're talking about areas without myelination, either unmyelinated axons or cell bodies and dendrites. Or if we're talking about myelinated axons in our white matter regions. Neurons will have at least one main axon. Some neurons will have more than one, and we'll talk about different kinds of neurons. But a main axon may give off branches, and these branches are called collaterals. This allows a single neuron to be able to communicate with multiple cells. And in cases of very long axons, such as an axon going from your brain all the way down in your spinal cord, it can send off branches to communicate with cells along the way. So it not only goes to a single target down in your spinal cord, but it might talk to other cells in the brainstem or other cells in the upper part of the spinal cord as it's going down to its final destination. At the end of each axon, as well as each collateral, there are branches called telodendria. And then the telodendria itself will end at an axon with axon terminals. So imagine that you have your body is an axon, your arms and legs are telodendria, and then each of those, you have your fingers and toes, those will be your axon terminals. And at the end of the axon terminals, so the end of your fingers, the tips of your fingers and the tips of your toes are going to be kind of enlarged to create an area for communication. And it is at these axon terminals, also called synaptic or presynaptic terminals, where the release of neurotransmitter takes place into a small gap between cells called the synaptic cleft. Axoplasmic transport is simply the transport of substances. It might be neurotransmitter, it might be nutrients, it might be some cellular components from the soma down along the axon to the axon terminal, or into a dendrite, or from the axon terminal to the soma. So it's basically all it is movement in the cytoplasm of substances to and from the soma. Anterograde transport means the movement of a substance away from the soma and towards an axon terminal. Retrograde transport is movement of a substance toward the soma from the synaptic terminal. Axoplasmic transport varies in rate from neuron to neuron. So it depends on the type of neuron you're talking about. But it does in all neurons have a tendency to be slower the older we get. So it takes longer to get neurotransmitter from the soma down to the axon terminal, where it can then be released to communicate. Okay, these axon terminals, as I said, they have kind of an enlarged ending, and these are called terminal buttons or terminal boutons or end bulb. So lots of different names, all for the same thing. The number of axon terminals will increase as learning occurs. So we not only increase dendrites and dendritic spines on the cell that's going to receive the information, we increase axon terminals on the cell that is providing the information. In addition to releasing the neurotransmitter into that little gap, the synaptic cleft, axon terminals can also reabsorb excessive neurotransmitter and break it down and reassemble. So certain types of neurotransmitters, as I mentioned, can be produced in the axon terminal. Our larger ones, our proteins and our peptides and some of the bigger molecules, they can only be produced, though, in the soma and have to be transported by axoplasmic anterograde transport to the terminal. Now let's talk about types of neurons. We have unipolar and pseudo-unipolar. So pseudo means false, so looks to be unipolar when really it isn't. We'll have bipolar, and we'll have multipolar. But first we're going to talk about the pseudo-unipolar. Unipolar, I'll mention, it has a single process. Usually these are interneurons. We don't really have any that I'm going to ask you to know. Okay, but pseudo-unipolar, very important, because our somatosensory neurons are pseudo-unipolar. A pseudo-unipolar neuron has two axons that come out of the soma. These axons, however, have fused together so that it looks like it's just one axon coming out of the soma that then splits in two or bifurcates. So originally there were two axons, but they fused themselves together to create the appearance of a single axon. And that's why it's a false unipolar, pseudo-unipolar. So those axons separate. One goes out to the periphery where it has dendrites on the end, and these dendrites have special receptor endings. The other goes into the central nervous system where it's going to synapse on another cell or cells within the central nervous system. The axon that is out in the periphery with the special receptor endings is called the peripheral axon. The axon that is going into the central nervous system is called the central axon. The peripheral axon conveys information from those specialized receptor cells towards the soma. That information then travels past the soma into the central axon and is carried into the central nervous system. Bipolar neurons are very small. They have two processes extending out from either side of the soma. So the soma is in the middle, and you have an appendage out one side and an appendage out the other. Typically, those would be the one appendage would be dendrite, and the other would be axon. Examples of bipolar cells are the retinal cells, the olfactory nerve cells for cranial nerve 1, and the vestibular cochlear cells within the vestibular cochlear ganglia for cranial nerve 8. Here we see these two different types of cells. So number 1 is a pseudo-unipolar cell. It looks like there's just a single appendage coming out of that cell body, and then that appendage splits in two, when actually they were separate and then they fused together by the cell body. You have the peripheral axon, which can be very long and is going out into the periphery. Somewhere might be the skin on your finger or the skin on your toe, or going into a muscle or a tendon or a ligament. It's got dendrites and with specialized endings that are sensory receptors of some type. That picks up sensory information. It sends it along the peripheral axon towards the cell body. It continues past the cell body into the central axon, which then ends with axon terminals in the central nervous system. Most cases, it'll be in the spinal cord, but not always. Sometimes it continues and goes right up into the brainstem. Number 2 is a bipolar neuron. And here you can see the difference, how there's an appendage on either side of the cell body, with the cell body stuck in the middle. And finally, we have multipolar neurons. Multipolar neurons have a single axon and multiple dendrites coming off of a variety of places on the soma. Some of these neurons have huge dendritic trees. Some of those in the cerebellum have been estimated to have 100 to 150,000 synapses on them because of these large dendritic trees. And there are certain neurons within the cerebral cortex that similarly have these huge dendritic trees associated with them. So multiple dendrites coming off of many parts of the soma, but a single axon. Where the axon comes off the cell body, there is an enlarged area called the axon hillock. This area has many ion channels in it, which will be important for the generation of action potentials. Voluntary motor neurons are one type of multipolar neuron, and many interneurons are also multipolar. Here we have a picture of a multipolar neuron. You can see the green area representing the cell body. Actually, where it's starting to come into the axon, that would be the axon hillock. You have dendrites coming off in multiple places. This would be a peripheral nerve since it's covered by Schwann cells for the myelin, forming the myelin sheath. Then we have the telodendria, and at the end of the telodendria we have axon terminals. The next image is another image that's maybe a little bit less cartoony, showing us the same thing. And this one you have a link to, so it's not actually in your file there, but your lecture note. But you have the link to this particular image. And again, you can see then the cell body. It shows some various organelles, such as Golgi apparatus and endoplasmic reticulum. Don't worry about those. But what I want you to see is the large number of dendrites with the dendritic branching on there. And there'd be spines coming off of those branches. So picture a spiny thorn bush. That's kind of what it's like. We have the axon hillock, where that axon is coming off of the cell body, so it's an enlarged area. Then the axon itself. This particular image does not show any myelin for the axon. That goes into telodendria, and then you can see at the end of each of the telodendria there are axon terminals that look like little buttons. So those are your terminal boutons or terminal buttons. And that's the end of our lecture on the various cells of the nervous system.